Look at the raw output of a modern DNA sequencer, and you won't see clear instructions or blueprints. You see a chaotic, infinite stream of basic biological building blocks, pure biological noise. We map genomes, train AI to predict protein structures, and design targeted therapies. But these advancements rely on a single shared currency: understanding biological sequences. Making sense of this genetic text requires forcing structural order onto the chaos. Researchers must line these sequences up and compare them, biological letter by biological letter. This computational method is known as multiple sequence alignment, or MSA. It is the process of arranging DNA, RNA, or protein sequences side by side to identify their exact structural similarities and differences. By mapping these similarities across multiple organisms simultaneously, biologists can trace exact evolutionary paths backward in time. This comparison reveals conserved regions. These are specific, highly stable stretches of genetic code that remain unchanged across species, signaling that they are essential for maintaining core biological functions. When a single biological entity diverges, splitting and mutating into distinct variations over millions of years, these conserved regions provide the constant reference points needed to track that lineage. Without multiple sequence alignment, the raw text of any genome is unreadable. Alignment is the necessary prerequisite for all comparative genomics and functional prediction. To execute this alignment, the bioinformatics community relies heavily on one specific tool, Clustal Omega, developed by the European Bioinformatics Institute. The software was built to solve a very specific problem of the modern era: the sudden explosion of high-throughput sequence data generated in genomics and proteomics over the last 2 decades. Aligning 3 or 4 sequences is mathematically simple. Aligning thousands of sequences simultaneously creates a massive computational bottleneck, demanding an algorithm that balances large-scale execution with pinpoint accuracy. Clustal Omega overcomes this bottleneck through a precise, deterministic algorithmic pipeline. This progressive architecture allows the software to process massive datasets while maintaining the accuracy required for downstream genomic analysis. The pipeline begins by ingesting the raw, unaligned biological data, whether that is DNA, RNA, or protein sequences provided directly by the researcher. The software cannot simply compile all of these sequences at once. To build a reliable structure, it first needs to evaluate the mathematical relationship between every possible individual pair. This animated diagram illustrates the first computational module, generating the distance matrix. The horizontal and vertical axes represent the individual biological sequences being analyzed. The algorithm calculates an initial pairwise similarity score for every single sequence provided. The intersection creates a specific numerical value representing the exact evolutionary distance between those 2 sequences. High similarity scores indicate a close evolutionary relationship, while lower scores reflect significant divergence. These values dynamically map out a complete web of relationships across the entire dataset. This matrix is agnostic to the type of biological data. It operates with the same exact mathematical rigor whether it is processing nucleotide or protein inputs. By generating the distance matrix, Clustal Omega translates raw biological text into a purely mathematical space, setting the stage for structural organization. However, a mathematical matrix is only a ledger of relationships. To move forward, this ledger must be converted into an actionable set of instructions for the software. This leads to the next phase of the pipeline, constructing the guide tree. The matrix is converted into a branching, hierarchical tree structure, known in biology as a dendrogram. The algorithm analyzes the ledger and clusters the most closely related sequences into tight, adjacent branches at the core of the tree. Conversely, more distant, divergent sequences are pushed to the outer branches of the hierarchy, visually representing their evolutionary distance. The guide tree establishes the precise order of operations, determining which sequences the software will merge first and which will be added as the alignment grows. With the order of operations established, Clustal Omega enters the execution phase, progressive alignment. The software follows the guide tree, starting from the center and working outward. It begins by aligning only the tightest, most highly similar clusters first. By aligning these closely related sequences, it creates a foundational block. This ensures that the most reliable, easily matched data anchors the rest of the process. The algorithm then moves progressively outward along the tree, continually adding more divergent sequences to the existing block while maintaining that core structure. This progressive strategy introduces a specific computational vulnerability. As increasingly distant sequences are added, the chance for alignment errors increases exponentially. To counter this vulnerability, Clustal Omega introduces a layer of precision through Hidden Markov Model, or HMM, profile techniques. HMMs are probabilistic models that overlay the progressive alignment. They calculate the strict statistical likelihood of specific biological sequences aligning at specific positions. These profiles act as a mathematical safety net, ensuring high structural precision even when interpreting complex evolutionary divergences. Combining the order of the guide tree with the statistical rigor of hidden Markov models is what allows Clustal Omega to maintain high accuracy without sacrificing speed. This chart shows a direct comparison between Clustal Omega and older legacy alignment tools across accuracy, scalability, speed, and large dataset handling. Older tools often lacked the computational architecture to handle thousands of sequences simultaneously, which resulted in severe processing bottlenecks and restricted scalability. By integrating HMMs into a highly optimized progressive pipeline, Clustal Omega allows researchers to process extensive genomic and proteomic data rapidly and reliably. By solving the scalability problem, Clustal Omega moved comparative genomics from a process limited by sequence count into a high-throughput scientific discipline. In the laboratory, the structural insights from sequence alignment guide the precise modification of proteins and the targeting of genetic sequences. In drug discovery and vaccine development, aligned sequences from Clustal Omega help researchers pinpoint target genes, informing the design of novel therapeutics. In protein engineering, locating stable, conserved regions allows scientists to reliably design custom proteins with specific desired attributes for use in medicine and industry. By analyzing genetic variations across wide alignments, researchers can support precision medicine initiatives, utilizing the data to tailor treatment plans to individual evolutionary traits. The deterministic output of Clustal Omega functions as the operational blueprint for modern biotechnology. Today, bioinformatics is heavily driven by artificial intelligence, from massive protein language models to advanced structure prediction tools like AlphaFold. This rapid advancement has created a common misconception that classical algorithmic pipelines like Clustal Omega have been rendered obsolete by machine learning. But AI modules are not magic. They are computational black boxes that require massive amounts of highly structured relational data to train effectively. Deterministic alignment tools act as the indispensable pre-processing backbone, providing the high-quality sequence relationships that AI models rely on for functional annotation. Artificial intelligence does not replace sequence alignment, it stands on its shoulders. Because of its open-source, deterministic architecture, Clustal Omega continues to provide the high-quality datasets that drive modern biological discovery.